Here it is. Well, just as I guess she was
So it didn't
wow, that one. I know. But now I know. I'm going to pull it up.
I'm listening to you.
So, Jesse wrote out I tried saying Jesse wrote out some real skills. And so Jesse found which jumper, which I don't think I'm proud that I just every day. Because it's private. Somebody else
All right, guys, welcome. The plan for today is we'll probably, I would imagine, get finished up with chapter 15 or close to finishing up with chapter 15. And that'll leave us 3 days for the endocrine system, right? We have Thursday and right? Okay, good. So 3 days for the endocrine system, which will give us plenty of time to really get into kind of the meat and potatoes of that system. And it's my favorite system in 227, so that'll be good. So what we've got on the plan for today and getting finished up with chapter 15 is we need to talk a little bit about the sense of equilibrium. So we talked last time we met about the ear and the structures within the ear that are related to hearing. I just briefly mentioned the structures within the ear that are responsible for the sense of equilibrium, so we'll be kind of building on that today. And then we'll also get into the sense of vision. So you guys at this point should have done the anatomy of the eye in lab, and so we're not going to be going over all of that anatomy. We'll kind of review some things, we'll kind of build on what you talked about in lab, but won't be going over all of that anatomy again. To help you understand the physiology is going to be our focus in here of how the eye actually works to bend light rays and how it works also to some extent in some detail to convert those light rays into electrical signals that are interpreted by the brain as visual scenes essentially. So that's kind of the plan for today, equilibrium and vision. I want to kind of remind you of what equilibrium is, because I think we're all aware of the fact that we see, that we hear, that we feel, that we taste, we smell. We're less aware of the idea that we have this sense of equilibrium that helps us to maintain posture and balance. And because of that, it was the last special sense that was actually discovered, and it wasn't discovered until about 100 years ago. It's something that functions in our body just kind of behind the scenes, and I don't think that we're really aware of it unless it's brought to our attention or unless we lose it, okay, which has happened to some people, and we'll talk about some case studies related to that as we go through this particular part of the lecture. So this is the sense that helps us to maintain posture, that helps us to maintain balance, that gives us information about the position of our body and where we're actually located in space. And there are 3 body parts that actually work together to help us to maintain equilibrium. The first of these are the eyes. And the reason that the eyes are so important in helping us to maintain posture and balance is they're giving us visual cues about the position of our body in space. For example, if I start to tilt, if I start to get a little bit off balance, if I can see properly, then my visual cue is going to feed that back to me that I'm actually off tilt a little bit, and I'm going to make some adjustments to that. So they're very, very important in helping us to maintain posture and balance, and they provide feedback that helps us with this particular special sense. Located in muscles, tendons, ligaments, joints, we have special neurons basically that are stretch receptors. The formal name for these are proprioreceptors, and they are involved in the special sense of equilibrium as well. And the reason for that is, in addition to when the position of our body changes, our visual feedback changes, we can tell that we're off kilt. We also get stretch, okay, and tendons, ligaments, joints, and muscles as we change position. So right now I'm standing pretty straight up and down. If I tilt off to the side, I'm going to start getting some stretch in muscles, tendons, and ligaments on this side of the body, and that information is going to feed back to me and help me also to know, in addition to the visual cues that I'm getting, that I'm a little bit off balance or I'm a little bit off tilt. In addition to the eyes and the proprioceptors, those stretch receptors that again are found in muscles, tendons, ligaments, and joints, there is a structure, and this is going to be kind of the focus of what we're talking about today, a structure that is located within the head that gives us specifically information about the position of the head and helps us to maintain equilibrium as well. And that's a structure located in the inner ear that we talked about a little bit the last time we met, the vestibular apparatus. So you can see the vestibular apparatus right here. It's the structure that's attached into the snail-shaped cochlea. It consists of the vestibule, which is basically a hollow chamber. We'll talk about its structure a little bit later on. And then the 3 semicircular canals, 2 of which are visible here. In lab this week, you guys did some experiments, or I guess it was last week now, did some experiments, or hopefully you did them, okay, looking at the sense of equilibrium and how these different things work together. So there was one experiment in lab where you had one of your lab group members stand at the board and then you drew, it's hard to talk for a long time to a lot of people. You drew lines, okay, outlining where their shoulders were, and then they had to stand there for 2 minutes and you watch them to see if they swayed back and forth. And usually what you'll see when somebody has their eyes open, okay, is they don't sway back and forth, and that's because the proprioceptors are giving information if they start to bend, if there's any stretch whatsoever. Okay, the vestibular apparatus, if your head starts to tilt, is going to give you back some information that you're moving. But in addition to those 2 things, the eyes, okay, the second you start to sway, give you that feedback that you're moving away from the midline in one direction or the other, and you very quickly make corrections to that. So you probably didn't notice much swaying, okay, they weren't going outside of the lines with their eyes open. But then the next thing that you did was you had them close their eyes, okay, and you again watched them for 2 minutes. And what did you guys notice with the eyes closed? Did swaying increase? Okay, what do you think would explain the increase in swaying when eyes were closed? Okay, your eyes aren't seeing and they're not making corrections. So when you have your eyes closed, proprioceptors are still working and the vestibular apparatus is still working, but you have eliminated this mechanism that also gives you feedback on position of the body. And so what happens is you see more swaying with eyes closed because you're not getting the immediate visual feedback, I'm off kilter. You're still getting feedback from the vestibular apparatus in the head. Okay, when the head's not straight up and down, you're getting feedback from proprioceptors in the body as they start to stretch, but you're going to get more sway because now you've got just those 2 things that you're relying on to maintain equilibrium, as opposed to all 3 of these things working together to give you that feedback. So that was kind of the reason for that experiment, to show you that there are those 3 things that work together to maintain equilibrium, and to also show you what can happen when one of those things is gone. So we saw in lab what happens when the eyes stop giving that visual feedback because you've closed your eyes and now you've got to rely on just the proprioceptors and the vestibular apparatus. There are case studies out there of people who have lost proprioception as well as people who have lost feedback from the vestibular apparatus. And in both of those cases, okay, if you've lost proprioception, now you're going to really have to overcompensate for that by using your eyes to kind of see where your body is in space, because you can no longer feel where it is in space. And you're going to have to watch with your eyes every single movement that you make. Same thing for the person that lost the vestibular apparatus and the feedback from there. It was actually a 93-year-old man, and he was walking around like this, okay, basically. And all of his friends were telling him, you're not straight up and down. Like, you were walking way off at a tilt. And he said, no, I'm not. If I was walking off at a tilt, I would definitely know it. I would be able to feel that. They finally convinced him to go see a neurologist. And so the neurologist who saw him was the man who wrote the book, the man who mistook his wife for a hat. This is another case study that's in that particular book. And the neurologist thought, okay, well, get up and let's see you walk. So this 93-year-old man, okay, walked through his office like that. And he said, you know, you are off to a tilt. And the man said, well, that's what my friends keep telling me, but I don't believe it because I would know if I was off to a tilt. And so the doctor said, okay, let's have you walk again, and I'm going to videotape you. So that's exactly what they did. He played the tape back for this little old man, and he said, oh my gosh, I am off to a tilt. What do you think my problem is? So they went through some testing, and what his problem was he was not receiving feedback from the vestibular apparatus. So he was not feeling that tilt of his head. This little old man had been a carpenter his whole life, and so he came up with his own solution to the problem. And he said to the doctor, so this vestibular apparatus is kind of like a level that's in my head, right? And the doctor said, yes, it tells you the position of your head so that you can make corrections to it when you are off kilter. And he said, I think I know what to do then. And this little old man wore glasses. He put a level in the rim of his glasses, okay, so that when he got off kilter, even though he couldn't feel it, he could see it, okay, and he could make adjustments using vision to get himself back up into position. So he had to compensate for losing this, okay, by focusing more on using his eyes to give him that feedback about the position of his body. There's another case study in that book, The Man Who Mistook His Wife for a Hat. This is about a girl who's in her 20s. She was well educated. This actually happened in the 70s. She had 2 little kids. She worked full time. There was nothing about her that would have seemed like she had psychological issues. But she needed to have her gallbladder removed. And back then, if you were having a surgery like that, you would go into the hospital a few days actually before, and they would put you on antibiotics and kind of do all of this pre-op care for surgery for a few days before the surgery. And so she went into the hospital for the first day, and she had this horrible dream that night that she wasn't able to feel her body. And she woke up the next morning, and that dream had come true. It probably happened in the night that she could no longer feel her body, and so it got put into her dream, essentially. That dream came true because she couldn't feel her body. Okay, she didn't know where her jaw was, and her mouth would just hang open. When she tried to walk, she would just collapse to the floor. She said that she felt like her legs were like 1,000 miles long, and she couldn't feel if they were beneath her. She couldn't feel if they were behind her, if they were in front of her. And if you can imagine, okay, not knowing where your legs are below you, not being able to feel them, that's going to make it very, very hard to walk. It's going to make it very, very difficult to even stand up. She really couldn't even sit up, because for us to be able to sit up, we need to be able to feel, okay, get feedback from stretch and muscles. If we're not getting that feedback, we could easily just do this, right? And without using our eyes or without relying heavily on our eyes, we may not know that we're slumping over in one direction or the other. So that's what had happened to this girl, and at first they just tried to tell her that it was a psychological issue, that she was nervous about her surgery, but the problem was that it never went away. And what they finally figured out was that she had had some inflammation in the spinal cord that completely took out impulses that were coming from proprioceptors. She had lost all of her proprioception. And so the way that she had to compensate for that was, again, using her eyes and being very active, okay, about watching movement. So for her to reach down and pick up a cup, she would have to watch her hand go to the cup. You know, she couldn't just do what most of us do, kind of half thinking about what we're actually doing in those movements, but she had to be very, very deliberate about it. When she was walking, she actually had to watch to make sure that her feet were below her, and this is what happened to her for the rest of her life. And she talks of an experience having had this happen of trying to get on a bus. And if you can imagine trying to get on a bus, okay, go up the stairs and find your seat and sit down without being able to feel where your body is. And of course, each step, she'd have to watch her hand to grab the handrail, and then watch her foot to get it below her on the stair, and then repeat that process. And somebody behind her was getting really impatient and said, are you handicapped, or are you drunk? You know, was basically the comment that person made. And her thoughts on that was, it's not like I can turn around and say, I just don't have any proprioception. You know, we don't know, nobody knows what that is, because we're so used to having it, we're so used to not living without it, that it's something that functions in our body really beyond even our sense of feeling it or knowing that it's there unless we really think about it and it's actually brought to our attention. What's thought to be responsible for car sickness, if you're somebody that suffers from that, is of course when you're sitting in a car, okay, the information that you're getting from these different structures is not meshing, okay? Your eyes are telling you as stuff is whizzing by you that you're moving. Your proprioceptors are telling you you're not moving, and your vestibular apparatus is telling you that you're not moving, okay, because you're not physically moving your body. And as those signals come in, okay, to the brain, and you're getting these mixed signals on movement or not, that is what seems to actually cause car sickness for a lot of people. So I had a student several semesters ago who had terrible car sickness. She even gets motion sickness on planes, which I thought was weird because it's not like you can really tell you're moving unless you're looking out the window, and then it seems like the movement's really, really slow based on perspective. And so she was going to McCall, and I told her, okay, get in the car, close your eyes, and don't open your eyes until you get to McCall. And I was like, she's not going to do that because that would be ridiculous. But she did, actually. And she said it was the first time ever that she didn't get car sickness because, again, she wasn't getting that input from her eyes that I'm moving while everything else was saying that she was stationary. That's why also if you get car sickness, it helps to sit in the front, because if you think about sitting in the back, okay, to look out the window, you're looking like this and stuff looks like it's whizzing by really quickly. When you're sitting in the front and you're looking forward, the perspective changes and it's not moving as fast. Okay? And that's why it helps to move to the front of the car if you're somebody that gets car sickness. Yeah.
I actually had sort of a situation like that. I had really bad dizziness that continued on for a few days, basically. And they found out that what had happened was I think there's like crystals or something in your semicircular canals, and they thought that some of them broke loose, and so it was throwing off my equilibrium. So even though I wasn't moving and like everything looked stationary, everything was going like this in my ear, and so it was throwing off my perception, and I was just having constant.
Totally, yeah, and that'll make you sick because again, you're getting that information that's jumbled. Yeah.
So how do medicine like Dramamine help with that?
What Dramamine does is it decreases the signal coming away from the structures that pick up movement. So it limits the action potentials that are sent away, essentially. So you're still getting those structures responding to movement, but you are less aware of it because the electrical signals aren't being sent to the brain at the same level that it would have been otherwise.
All right. And is there any specific reason why motion sickness comes more as an adult than when we were kids, per se?
Not that I know of, but I totally understand because I used to just spend all day and swing, and now the thought of that makes me bummer. Anything else?
So when people get vertigo, is that a problem with one of these three areas?
It is. Usually it's a problem with the structure right here. Anything else? All right. So we've had kind of a good deep introduction into what the sense of equilibrium is and the different structures within the body that allow us to sense our posture, position of the body, help us to be able to maintain balance. What I'm going to do now that we've had kind of that general introduction is get more into the specifics. So there are actually two different types of equilibrium. We'll talk about the two different types, and we'll look at the structures within the inner ear that are responsible for detecting each of these different types of movements and allowing us to maintain equilibrium. And then we'll also talk about functionally how they work once we get done with the anatomy. So the first type of equilibrium is what's known as static equilibrium. And the receptor organs for static equilibrium are located within the vestibule, so that hollow chamber that sits between the cochlea and the semicircular canals. And what static equilibrium does, or what these receptors that help us to maintain static equilibrium do, is they detect linear movement of the head. So by linear movement, I mean essentially movement in a single plane. If you are moving up or down in an elevator, that sense of movement that you get as you're moving up or down is detected by static equilibrium. If you're moving forward, okay, or backwards, maybe on a train, okay, that sense of movement that you get either forward or backwards is detected by static equilibrium. When you move side to side, okay, but again in a single plane, a side-to-side movement, that's also a movement that's detected by static equilibrium. The other type of equilibrium is what's known as dynamic equilibrium, and the receptors for this particular type of equilibrium also are located within the inner ear, and it's the semicircular canals actually that contain the receptors for dynamic equilibrium. Dynamic equilibrium detects rotational movement of the head, so movement that is not just in a single plane, but movement that is more like a 360-degree movement. So if you can imagine like a figure skater, okay, spinning, or a ballerina spinning, rocking back and forth on a boat, that's not just a front to back or an up and down movement, but it kind of combines those two movements into more of a rotational movement. Shaking your head no, okay, is a rotational movement. Shaking your head yes, okay, because you're moving forward, and also up and down is a rotational movement. Tilting your head is a rotational movement as well. So those types of movement that aren't just movement in a single plane, but kind of combined movement in two or more planes essentially, is rotational movement, and that's what dynamic equilibrium monitors and responds to help us be aware of the position of our body and also to help us maintain posture and balance. So with that introduction, what I want to do is talk a little bit about the anatomy of the receptors for static equilibrium and how it actually works. And then we'll talk also a little bit about the anatomy of the receptors for dynamic equilibrium and how that special sense works as well. So you guys should be familiar at this point with seeing the structures within the inner ear itself. Right here is where we have the structures that are involved in both static equilibrium and dynamic equilibrium. As I said previously, the receptors specifically for static equilibrium to be able to detect that linear movement, okay, movement in a single plane, are located here within the vestibule. And as I mentioned before, the vestibule essentially is just a hollow chamber. So if you look at this picture of the structures of the inner ear kind of separated out a little bit more highly magnified so you can see the details. Again, it's right here in this area of the vestibule where we actually have those receptors for static equilibrium. And there are basically two hollow chambers within the vestibule itself. The first of these is known as the utricle. You can see it right here. The second hollow chamber is what's known as the saccule, and you can see that structure right here. And what I want to do is focus your attention to a little patch. It's represented in green on each of these diagrams, and this patch is known as the macula, and it's actually within the macula itself that we have the receptors for static equilibrium. So what we're going to do as we go to the next diagram is we're going to be focusing on what the macula actually looks like. Again, this structure within the vestibule, there are two of them that actually contain the receptors for maintaining equilibrium. So if you look at this picture up here, again, we've got the vestibule. There's those two hollow chambers, the utricle and the saccule that I mentioned before. And it's within the utricle and within the saccula that we have this little patch or this little area which is known as the macula that actually contains the receptors for equilibrium. If you look at this picture down here, what we've done is we've taken the macula of the utricle, we've magnified it quite a bit more highly. Okay, so you can see the detail of the structures and you can see the detail of the cells that are located in this area that allow us to be able to detect static equilibrium. So I want to just point out to you some of the unique anatomy that's associated with the macula. And again, there are two of these. Okay, the first thing is we've got some neurons which are known as hair cells. And again, the reason that they're known as hair cells is they have these little hair-like extensions that branch off of their dendritic end, which is so commonly seen with the special senses. So again, we refer to them as hair cells, and these little hairs that extend off of them are the stereocilia. If you look at the stereocilia within the macula, you'll notice that in each little patch of stereocilia, there's one that's modified. Okay, it looks a little bit different from the others. And this modified stereocilium that we see in each of these little clusters of stereocilia branching off a hair cell is what's known as the kinocilium. Okay, and the kinocilium is modified. It's important for detecting the direction of movement. So we'll talk about it a little bit later on. But again, it's just a modified stereocilium that's important for detecting the direction of movement, okay, when you have linear movement. For example, is the movement forward? Is the movement backwards? Is the movement to the right side? Is the movement to the left side? Is the movement up or is the movement down? Would be the specific information that the kinocilium would be able to give you. Sitting atop this patch, okay, the macula, we have kind of a gelatinous membrane. It's represented in white. This gelatinous membrane is known as the otolithic membrane. It's going to add some weight to the hair cells, essentially depress those hair cells a little bit. The reason that this structure is known as the otolithic membrane is because it contains little rocks, okay, little crystals of calcium carbonate, which we refer to as otoliths. So if you've had medical terminology, you know that what that literally translates to is ear rocks, okay? So these are little crystals of calcium carbonate. Calcium carbonate is what seashells is made out of. It's what chalk actually is made out of as well. So little crystals, little rock-like structures of calcium carbonate that sit atop this membrane and again are going to give some weight to it. Before we get into, so we've got the anatomy down, it's really kind of simple, the anatomy of the macula. Before we get into the physiology, I want you to look at, okay, the macula here in the utricle and the macula here in the saccule, and notice that they are oriented differently. So if you look at the macula here in the utricle, you'll notice that basically it's oriented horizontally. So if you could imagine, okay, the arm as the macula and my fingers as the hair cells are going to be standing up in this direction, okay, within the utricle. If you look at how the macula is oriented in the saccule, you'll notice that it's up and down. So again, if we use the same analogy, my arm is the macula in the saccule, okay, it's oriented like this, and the hair cells are extending out in this direction, okay. As you think about that, any ideas why we need two and why they need to be oriented in different planes? Okay, so you guys are talking about being able to detect different movements. Let's look at the macula within the utricle. And I know we haven't talked exactly about how they detect movement, but maybe you can kind of think about how they might. The macula in the utricle, again, is oriented like this with the little stereocilia, okay, extending up like this. What allows the macula of the utricle to detect movement is when those stereocilia get bent in one direction or another, okay? So if we have a macula that's oriented horizontally like this, what type of movements is going to be detected by this macula? So here's your choices: up, down, side to side, front to back. Okay, which of those would not be detected by a macula that's oriented like this? Up, down, okay? Because what's going to happen is when you move front to back with that otolithic membrane sitting atop the stereocilia, they're going to get bent, okay, in one direction or the other. As you move side to side, they're going to get bent, okay, by that side to side movement. So what we've got going on in the utricle, and this is easy to decipher, again, just based on the way that it is oriented, is it's going to be able to detect the side-to-side movement. It's going to be able to detect the front-to-back movement. If you look at the saccule, oriented completely differently, again, it's up and down, it's vertical, so that when you have upwards movement or you have downwards movement, that's what's going to cause the bending of the stereocilia in that particular area. So just something to think about and keep in mind is the way that these things are positioned and how, based on their position, that's going to differ as far as the types of movements that they're capable of responding to. So you guys have kind of already deciphered this out, or many of you, based on the answers that you gave to that last question. But I'll go ahead and go over this formally, how these structures, the macula, or the two macula within the vestibule are working to be able to detect linear movements. And here's a nice picture that kind of represents this for us. So we've got the macula here, hair cells with their stereocilia projecting up. Here's the otolithic membrane containing those otoliths. And if you look at what happens as this person slumps their head forward, okay, we've got a linear movement in a forward direction. What's going to happen is inertia is going to cause the otolithic membrane in that case to slide forward essentially. As the otolithic membrane slides forward, it's going to cause bending of the stereocilia. Now, if you were to slide your head backwards instead of forward, of course the stereocilia would be bent in the opposite direction. So we get the stereocilia being bent as this otolithic membrane slides either forwards or backwards. Bending of the stereocilia towards the kinocilium. Okay, remember that little modified stereocilium that we talked about previously. Bending of stereocilia towards the kinocilium causes a different situation than bending of the stereocilia away from the kinocilium. So that's how we're able to detect, is it forward movement or is it backwards movement, is which direction those stereocilia are being bent essentially. Is it towards that modified hair-like structure? Is it away from that modified hair-like structure essentially is the idea. There's kind of an interesting situation that occurs with these hair cells. So what we've been taught up to this point is you get a change in a neuron's environment, and that causes it to generate an action potential, and that action potential causes it to release neurotransmitter at its synaptic end, okay, somewhere where it synapses with another neuron. It's kind of different with these particular cells here. They are actually always depolarizing and always releasing neurotransmitter. Okay, so from the get-go, we're a little bit different with this particular situation. Bending of the stereocilia towards the kinocilium causes these hair cells to depolarize and release even more neurotransmitter. So they're always releasing it, but when it's bent towards the kinocilium, we release more. Bending of stereocilia away from the kinocilium causes the hair cells to hyperpolarize, and they start releasing less neurotransmitter. Okay? So whether the bending happens towards the kinocilium or away from the kinocilium, we get a change in the electrical signal. In some cases, it's an increase in the amount of neurotransmitter that's being released. In the case where we're moving away from the kinocilium, it's a decrease in the amount of neurotransmitter that's being released. But either way, okay, the brain can interpret that as a change in the position of the head. And based on whether it's more neurotransmitter being released or less, it's going to tell us, is it front or is it back? Okay, is it to the right or is it to the left? Is it up, is it down, essentially, based on the way that that electrical signal is actually changing.
So the question is there a situation in which, say there's defect in the cilia so that somebody feels as if their head is, straight up and down when it's actually tilted, so that a person will always have their head tilted like up, down, side.
There absolutely could be, and that's exactly what happened to that man, and the man who mistook his wife for a hat is his stereocilia weren't responding. He had nerve damage or he had stereocilia damage, whatever happened, his vestibular apparatus was not working like it should to give him that feedback, and so he thought that this was straight up and down because he couldn't feel a difference when it absolutely was not, so definitely yes. Yeah?
So these stereocilia, they won't break off because they don't have a plate rubbing against them, but what would something like whiplash do to them?
They won't what?
Break off because they don't have a plate rubbing against them, like the ones in the ear?
So they do. They have the otolithic membrane that slides back and forth over them. It's more gelatinous. It's not as hard as the textural membrane in the ear. I don't know what whiplash will do to them. Has anyone in here had whiplash? I never have. I don't know if you would get some nausea associated with that, and if you did, it could be related to some temporary damage in there. I just don't know what. Oh, sorry. Anything else?
So you said that they are constantly releasing neurotransmitter, but are they creating action potentials when they do that?
They are. So the action potential traveling down that neuron is what would lead to the neurotransmitter being released. So these neurons are always depolarized, always sending action potentials, always releasing neurotransmitter, and we can up the frequency of that signal or we can decrease the frequency of that signal, essentially is what's going to happen. But either way, the brain is going to read that as movement in one direction or the other. Based on the way that the frequency changes, that's how it's going to know if it was forward or was it backwards. Was it to the right? Was it specifically to the left? Was it up? Was it down, essentially? Anything else? Okay. So we finished up talking a little bit about static equilibrium, and I mentioned I also want to talk about dynamic equilibrium as well. The receptors that allow us to be able to detect rotational movement of the head specifically. So again, that's movement that combines more than 2 or more planes, essentially, movement in 2 or more planes. Those structures are also located within the inner ear. Specifically, it's the 3 semicircular canals that are responding to rotational movement. There are 3 of them. Okay, I'm not concerned that you need to be able to identify them. I want you to know that there are 3 and their names. They are very small. Okay, so if you were to look at the circumference of these semicircular canals, it would be equivalent to about the eraser on the end of your pencil. So each of those little circular structures would be about that size, essentially. They lie in 3 different planes. Okay, so you can kind of see that here, maybe, if you use your imagination a little bit. And the reason that they lie in 3 different planes is so that they can detect rotational movement in all 3 planes. So one of them, okay, is detecting this type of rotational movements where you're combining forward and up and down. One is detecting this type of rotational movement, okay, where you're combining the side-to-side movements and also kind of rotating the head. It's not just a front to back or left to right. One of them is detecting this type of rotational movement, okay? And so we can feel if we're rocking back and forth on a boat, we can feel when we're spinning around or when you're swinging by the signals that are coming from these different structures within the inner ear. What I'm going to do is look at the structure of a semicircular canal. So this gives us kind of the big picture here. Here's one of those 3 semicircular canals represented for us, and all of them have the same anatomy, they have the same structure, they just differ in the plane that they're oriented in, essentially. So they all respond the same way, but they're able to detect rotational movement in different planes because of the way that they're actually oriented. If you look at a semicircular canal, the first thing that I want you to notice is here at its base, it has kind of a little swollen portion to it. This swollen portion is known as the ampulla, and it is within the ampulla that we have the structures that actually respond to dynamic equilibrium. So if you look right here, we've got kind of this little mushroom-shaped colored structure that sits inside the ampulla, and then the artist is representing that as being magnified much more highly over here, so we can see the detail of the cells and the structures that sit within the ampulla and allow us to respond to rotational movement. If you look at this picture, okay, this structure within the ampulla as a whole is referred to as the Crista Ancillaries. So the Crista Ancillaries essentially is the receptor for dynamic equilibrium. The Crista Ancillaries is composed of what's known as the cupula. So this is represented in purple. It's just a gelatinous mass. And the cupula sits atop some hair cells, okay, which in this diagram are represented in blue. And you'll notice, as we've seen before with the special senses, as is so common with the special senses, that these hair cells have these little hair-like extensions that extend off of them and up underneath the cupula, essentially. The other thing that I want to say, in kind of giving you an understanding of the anatomy of the semicircular canals, is that they are filled with fluid. Okay? And so as you're moving your head in a rotational plane, that's going to cause movement of this fluid in the semicircular canals, essentially in one direction or another. So with that, okay, we're set up to be able to understand how the structure actually works within the ear to be able to detect rotational movement. And so I want to kind of formally walk through how that process works with the next couple of slides. The first thing is the head moves in a rotational plane. Okay? And when the head moves in a rotational plane, fluid in the semicircular canals is going to start to move as well. And that fluid is initially actually going to oppose the movement of the semicircular canals. So if you like to go to the fair and ride that Viking ship, okay, that does this, as you are sitting up here and you're getting ready to come down, your semicircular canals are moving in that direction, but what direction is the fluid actually going to move? Okay, it's going to move in the opposite direction. So you're going to get that fluid initially opposing, okay, the movement of the semicircular canals. Well, when that happens, okay, we've got this little gelatinous structure in there. This fluid is moving over the gelatinous structure, and that's going to cause that entire gelatinous structure to bend. Okay, so if we're moving this way, the gelatinous structure is going to be bent in that way. If we're moving this way in a rotational plane, the gelatinous structure is going to be bent in this way, okay, as the fluid initially opposes that movement. But again, with the same thing that we saw with the macula, as we get the cupula moving back and forth, okay, kind of analogous to the otolithic membrane, that's going to cause bending of the stereocilia in one direction or the other. Okay. What we've got going on with these hair cells here is the same situation that we had with the hair cells in the macula. So they're constantly depolarizing, they're constantly releasing neurotransmitter, depending on the direction that the cupula is bent. Okay, so depending on the direction that the stereocilia are bent, these nerve cells are either going to depolarize and release even more neurotransmitter, or they're going to hyperpolarize and release even less neurotransmitter. Either way, okay, we're going to get a signal being sent to the brain that there's rotational movement that's occurring. Whether that's a hyperpolarization or a depolarization is going to allow the brain to determine in which direction that rotational movement is occurring. So very, very similar to what we see with static equilibrium and bending of the hair cells in the macula, but this allows us to detect rotational movement because of the shape of the semicircular canals and because of the fact that they lie in those three different planes essentially.
So when somebody gets like seasick, usually it's people that don't go on boats very often. Okay, then you have people who are, say, fishermen who are always on boats, and they don't get seasick at all, no matter how rough it basically gets. So is it just a matter of they're overcoming the feeling, or is it something that gets changed over time that makes it easier? Or?
So I think there's a variety of things. One of these things is a situation which is known as central adaptation. Central adaptation is something that we would have talked about in chapter 13 had we covered that chapter. But what happens with central adaptation, and you guys did some experiments in lab that kind of showed this, for example, when you put the pennies on the arm, and you initially felt that very strongly, but then over time you adapted to it, so that sensation went away. That's central adaptation. And so I think there's some of that that comes into play. Our brains actually throw out about 99% of the sensory information that's coming into us, because if they didn't, we would go absolutely crazy, because I would constantly feel my hair in a ponytail and my hair being pulled, and I would feel these things dangling around, and I would feel my wedding ring, and I would go insane, you know, and I would feel the band around my pants, and that would be irritating. We all have these things that if we weren't capable of just kind of weeding that stuff out, that it would drive us insane. So I think part of it's that. I think, and I haven't spent a lot of time on a boat, and I do get very seasick, I think part of it is learning how to adjust to the movements of the boat, so you're not getting thrown all the time, and then you're not moving as much, so you're really not getting as much signals being sent back. And I think part of it's psychological too, because if I even think about going on a boat, I'm like, oh gosh, you know, and before it even starts, I'm already just miserable. So I think there's different things that come into play.
So as far as like the movement, you think that the body adjusts and the head stays more still?
I do. I think you kind of get your sea legs and you know how to keep this part of you still and just kind of move with the boat. Would you say yes? Okay,
Yeah.
or sailor.
When we pull into port, we'd leave the ship and everybody would walk down the pier. All funky. I mean, it just, until we got used to the fact that the world was not moving,
Did you
anymore.
start to feel seasick again once you
I
first
never,
got back
I've
to
been through 2 typhoons and a hurricane and never felt seasick.
What do you mean, like when you first get back on the boat, because you get the opposite effect where you feel like that.
The only time is right after it leaves the pier.
That's what I'm talking about.
You immediately know that you're not. It moves it moves on the pier. It goes this way, but then it starts going this way when you're out to sea.
Watching Navy soldiers coming back from trips out is the funniest thing.
Yeah, because we walk.
Okay, so I don't know if you all can hear that, but the question was about how do you adjust to that and how do you not get so sick? And so from our sailor and ex-military people, so there is some stuff with adaptation, but you also learn how to walk so that your head's not being thrown every direction as you're on a boat. So yeah, all that stuff comes into play as well. Anything else? Okay. So we'll switch over to the eye. It looks like we have about half an hour left. That may or may not be enough time for us to get through the eye, but we'll do what we can. The eyes, of course, are the organs of sight. And I want to start out with talking a little bit about some of the accessory structures of the eye. These may or may not have been things that you talked about in lab. I typically don't, just because we don't have models for them, and so I don't focus on this anatomy. But I've got several accessory structures to the eye listed here. Things that help the eye to perform its particular function involved in the sense of sight. And the first of these that I want to mention is a structure which is known as the lacrimal apparatus. So if you look at this picture up here, the lacrimal apparatus are the organs that are involved in producing tears. Tears are important for moisturizing the eye, keeping it moist so it can work properly, so it doesn't get inflamed. It's also important for cleansing the eyes as well. So if you look up here, these structures are all a part of the lacrimal apparatus. This is the lacrimal gland. It's what actually produces the tears. As tears are produced by the lacrimal gland, they wash across the eye in this direction to moisturize the eye, to wash anything out of the eye that may be in it. And then those tears actually get drained into what's known as the nasolacrimal duct, which you can see right here, that actually runs into the nose. So the tears actually drain into the nose itself. It's not known why the lacrimal gland becomes more active when we're emotionally upset, but we all know that it does, and we start to cry, and we start to produce so many tears that they can't all drain through the nasolacrimal duct, and those tears start to overfall and run down the cheeks. But that's one of the reasons after you've been crying that you get a runny nose, okay, because you had an increase in tear production and an increase in this watery secretions down into the nose itself, because that's where the tears drain. And that's what makes the watery, stuffy nose when you have been crying. For those of you that get allergies, okay, seasonal allergies, and you know that you start to get the runny, watery, itchy eyes, that's because there's inflammation in this entire area. A lot of times the inflammation will partially close the nasolacrimal duct, and that will prevent tears from being able to drain down into the nose. And so instead it'll seem like your eyes are watering. Really they're probably not producing a whole lot more tears than they would normally. They're just not draining properly because of the swelling and the inflammation closing off the nasolacrimal duct. So again, primary function just there to cleanse the eye, to keep it moist so that it can work properly. Eyelids, also an accessory structure. The eyelids contain the thinnest skin in the body. They are, of course, protective to the eyes, so when we blink, okay, that helps to spread those secretions from the lacrimal gland across the eye. When we blink, that can help to protect the eye, and we reflexively blink, of course, when something comes near the eye. Eyelashes, also protective, also one of these accessory structures. You guys had a chance in lab, maybe some of you did it, to test the corneal reflex a few weeks ago. And one of the things that you noticed, of course, is when something's coming at the eye, you tend to blink reflexively. That's meant to protect the eye, to prevent that thing from becoming lodged in the eye. If you look at the eyelashes themselves, they're very highly innervated, and typically when something touches the eyelashes, okay, that causes reflexive blinking as well, because that would indicate that there's something close to the eye. We don't want that thing, whatever it is, getting into the eye, and so it makes sense to have these hairs very, very highly innervated and be tied into this reflex that causes blinking to occur, just to protect the eye itself. Eyebrows, believe it or not, also protective. They help to shade the eyes to some extent from UV rays. The other thing is we have a very, very high density of sweat glands in our forehead. It's one of the areas of skin where sweat glands are found in the highest density. And so the other thing that eyebrows do to some extent is when we start to sweat heavily, they prevent that sweat from running into the eye. It acts as a physical barrier to prevent sweat from getting into the eye itself. Last accessory structure that I wanted to mention is a very thin membrane, which is known as the conjunctiva. If you look at this picture down here, you can see the conjunctiva represented nicely. It actually lines the eyelids, so the inner portion of the eyelids, and then it folds over and it aligns the entire exposed portion of the eye. And then you can see that it folds over again and it lines the upper eyelid as well. If you look right here, it's been partially torn away, just I think so that you can picture in your mind that there is a thin membrane that's sitting over this entire exposed portion of the eye, and that's also lining the eyebrows as well. Pink eye, okay, is actually conjunctivitis, which is just inflammation of this membrane. A lot of times it's caused by allergies. And just the inflammation of this entire area when you have hay fever, when you have seasonal allergies. It can also be caused by viruses or a bacterium in the eye. Okay, so not washing your hands after using the bathroom or something like that that would cause those types of things to get into the eye can lead to it as well. This particular structure, if you wear contacts, okay, you know sometimes the contact gets off the iris of the eye, the colored portion where it should be, and it goes like up under your eyelid. Sometimes people worry, is it going to go entirely behind my eye? It can't, and that's because the conjunctiva folds over. So if you have worn contacts and you know sometimes you lose it, it gets stuck up there somewhere. It just goes and sits under the eyelid and it can't move any further because of the conjunctiva preventing it from actually going back behind the eye itself. So we're going to do just a little teeny bit of anatomy, probably some review from what you guys actually talked about in your lab classes, just to make sure that we're all on the same page as we move forward and we have an understanding of these structures so that we can understand physiologically how they work. The first thing that you probably talked about in lab is the fact that the eye itself has 3 different layers to it. So if you can imagine taking an eyeball out, okay, cutting it like this, and then looking down in on the internal structures of the eye, that's the perspective that we're getting here essentially. The outermost layer of the eye is thick, it's dense, it is sturdy, and it is protective. And it is known as the fibrous layer. So if you look at the eye here, okay, these areas that we're seeing in white, again, thick, dense, sturdy, protective, those are the areas of the fibrous layer. You guys learned in lab, or I hope you learned, that the fibrous layer has 2 different components to it. So we've got the sclera, and the sclera is composed of connective tissue. It's on the posterior, okay, of the eye. When you look at somebody, the whites of the eye that you're seeing are the sclera. And the other portion of the fibrous layer is what's known as the cornea. So the cornea is just this most anterior part of the fibrous layer. You'll notice that it has this convex shape to it, okay, so it's bent out at an angle. Because of its convex shape and the fact that it's translucent, it's completely clear, okay, unless it has damage or injury or something like that. Because of that, it allows light rays to pass through it because it's completely translucent. But the other thing is, and we're going to get into this a little bit later on as we start talking about some physics and light actually passing through surfaces. The other thing is, as light rays pass through the cornea, they're actually bent by the cornea and they converge. So the cornea actually works as a lens within the eye, and it is actually the more powerful lens in the eye. It focuses light rays, it converges light rays more strongly than the lens itself actually does. So it works with the lens to help us to be able to see, to converge light rays so that we can see properly. So more about that a little bit later on. I just wanted to throw that out there while we're actually looking at its anatomy and looking at the shape of the cornea itself. Second layer of the eye wall is what's known as the vascular layer, sometimes also referred to as the uvea. So in this diagram, it's all of these areas that are represented in red. It actually consists of some structures that collectively are known as the vascular layer. One of these is the choroid. If you remember that term from lab, the ciliary body or ciliary muscle is a part of this vascular layer, and the iris is as well. Okay, so these three structures that you discussed in lab are all a part of the uvea, or all a part of the vascular layer that's represented in red in this particular diagram. The last layer of the eye, and this is the only one that we're going to kind of focus in on the anatomy, because this is where kind of the physiology of the eye is happening as far as converting light rays into electrical signals. The third layer, the innermost layer, is what's known as the sensory layer. It's represented in yellow on this particular diagram. One of the things about this layer is it contains within it neurons that are known as photoreceptors. Okay? Photoreceptors are a special type of neuron that specifically are stimulated or specifically respond to changes in light intensity. So we've talked about chemoreceptors and the fact that they respond to chemicals in solution. We talked about proprioreceptors, which are stretch receptors. They respond to stretch. What these receptors are detecting, located here in the sensory layer of the eye, are changes in light intensity, and that allows us to see the things that are around us. What we're going to do next is go to another slide that kind of focuses in on this innermost layer, on the sensory layer, and gives us some detail to the structure of the sensory layer itself. So I'm going to start out with talking about this diagram down here. If you look, here's the whole eye, same perspective that we were seeing before. The artist has taken just a small section of the back of the eye and magnified it much more highly for us so we can kind of see the details. And what I want you to notice, if you can look at the details here, is here we've got that sensory layer. Okay, it consists of several different cell types that are all oriented together. And what happens as light comes into the eye is light enters through the cornea, it enters through the lens. The cornea and the lens work to converge those light rays. The light rays are focused, okay, here on the sensory layer, and it causes these photoreceptors within the sensory layer to depolarize, okay, and to start to send action potentials and electrical signals away from them. If you look at the sensory layer itself, it's also known as the retina, and it has a couple of different sections to it. So if we take just this section here, okay, just this part of the back of the eye focusing on the sensory layer, and we magnify it a little bit more highly, you can actually see these two layers of the retina and see how the cells are oriented, and that will allow us a little bit later on to talk about how they actually work. Okay? So here is the retina, or the sensory layer. This is the most posterior part of it here, and what we've got in this most posterior part of the retina is the first layer, which is known as the pigmented layer. This is a layer that works to support and to nourish the photoreceptors. Yeah. The pigmented layer is a layer that supports and nourishes the photoreceptors. If we go just anterior to the pigmented layer, you can see that we've got a layer of neurons here. Some of them are represented in purple, some of them are represented in yellow. These are the actual photoreceptors. Okay? we've got two different types of photoreceptors in this first layer of cells. The first is known as rods. The reason that they are known as rods, and I've got some pictures of real cells of this type a little bit later on so you can see this, but the reason that they're known as rods is because they have a rod shaped into them essentially. So as you look at this diagram here, our rods are the photoreceptors that are represented in purple, and you'll notice that their shape is very much rod-like, okay, or they have a rod shape to them. Rods, as you may have talked about in your lab classes, are hundreds of times more sensitive to light than are the other photoreceptors, the cones. And because they are hundreds of times more sensitive to light, they can be stimulated by very, very low light levels. And so they allow us to be able to see in dim light conditions or in low light conditions. And that's actually how I remember them. Rod ends with a D, dim starts with a D. And so if I can just keep that in mind, that helps me to remember that rods allow us to see in dim light. You may have noticed when you're in dim light situations that you see grayscale. So you don't see color when you're out walking at night. You don't see color when you get up at night to go to the bathroom, or when you're wandering around at night to get a drink. Because rods do not allow us to have color vision. They do not give us sharp, acute vision either, and I think that's why sometimes we're laying in bed, we hear a noise, we see our coat draped over our chair, and that looks like somebody hunched over, because we don't see as acutely and we don't see in color with the rods when they're activated. But they are much more sensitive to light, and that allows them to be stimulated by very, very low light levels, and allows us to have some night vision to some extent. The other type of photoreceptor is what's known as a cone. Cones are not as sensitive to light. They require very bright daylight intensity light to be activated. But cones, when they are activated, provide very sharp, acute vision, and they also allow us to see in color. And that's how I kind of remember what they do, because cone starts with a C and color starts with a C, and that helps me to kind of keep them straight. If you go back to the diagram that we were talking about previously, the reason that cones are known as cones is because if you look at their shape, they're represented in yellow on this diagram. If you look at their shape, they have kind of a pointy tip to them, or a pointy cone-shaped end, and that's just where they got their name, is from their actual shape. So what happens when light enters the eye is it passes through the cornea and the lens, that focuses it down on the retina. It causes the photoreceptors, the rods and the cones, to be stimulated. Okay? The rods and cones send signals as they're stimulated to the second layer of cells. You can see we've got some additional neurons, a second layer of neurons that are represented in red. These are known as bipolar cells. That causes them to be stimulated. They send signals to the third layer of cells that's within the neural layer, which are the ganglion cells. Ganglion cells, as you can see right here, feed into the optic nerve. So we basically have got this situation going on with light entering the eye that seems almost a little bit counterintuitive, where light enters, it passes through all of these structures to the most posterior layer of cells, which is the rods and cones, causes the rods and cones to be stimulated, which causes the bipolar cells to be stimulated, which causes the ganglion cells to be stimulated, which sends signals into the optic nerve. So our pathway of light is opposite the pathway of electrical signals that's actually leaving the back of the eye and going to the brain to be interpreted. So there's some additional things that I want to talk about with regards to the sensory layer itself, some other structures that are associated with it. The first of these is what's known as the optic disc. It's also sometimes just referred to as the blind spot. The optic disc is this area of the sensory layer right here, where all of these nerve fibers from other areas of the retina are coming together to form the optic nerve, and that optic nerve is exiting the back of the eye. The reason that this is also known as the blind spot is because this is an area where there are no rods and cones. And since there are no rods and cones, there are no photoreceptor cells to be able to pick up light rays that are landing on the eye here. So So what happens as light enters the eye, okay, is it's focused down through the cornea, through the retina, and then it lands, okay, on the back of the eye. But it doesn't land on just one area of the back of the eye. Light rays, okay, from different areas in our visual perspective are coming in, and they're landing on specific areas of the retina itself. The light rays that are landing in this particular area are light rays that are coming in from images out in our peripheral vision. Okay, so they come in, they move through the cornea, the lens, and those light rays get projected right here. Well, there's nothing to pick up those light rays. There's nothing to be able to detect those visual images from those areas because there's no rods and cones. And that's why last week in lab you guys had the chance to do that experiment where you looked for the blind spot and you took your lab book and you slowly brought it close to you, and when the X disappeared, it was in that area where those light rays are being transferred into the optic disc. There's no way to pick them up, and so that X completely disappeared. Now, we're not aware of this big area that we can't see anything in out here in our peripheral vision on either side because our brain is very, very good at just kind of filling in the information and just kind of putting something there. So we don't recognize that there's nothing out here in the peripheral vision where we have the blind spot. The other thing that I wanted to mention that is associated with the sensory layer, and a good thing for you to have an understanding of, is a structure which is known as the Macula Lutea. If you look right here, here's that optic disc, okay, where the optic nerve is exiting out of the back of the eye. You can see that everything kind of enters and exits into the retina from that area. If you go just lateral, okay, to the optic disc, where that nerve is exiting out of the back of the eye, there is a structure which is known as the Macula Lutea. The Macula Lutea is an area where there's a very high density of cones. In fact, about 50% of the nerve fibers in the optic nerve are coming specifically from just the area of the macula. Within the macula is a very, very small pit, maybe only about 1 millimeter across, which is known as the fovea centralis. So you can see the fovea centralis there, and the fovea is the area of highest cone density. So there are more photoreceptors in that area than in any other area on the back of the eye. With that having been said, as you think about light coming into the eye and then landing on all of these different areas of the retina, we've already talked about the fact that the light that lands right here isn't being picked up. We're not able to get any images from that light because there's no photoreceptors there. What about images that are landing here? How well are we going to be able to see them, since that's our area of highest cone density and where 50% of the nerve fibers in the optic nerve are actually coming from? How's our visual acuity going to be here? Awesome. Okay. It's going to be really good. How's our visual acuity going to be in all of these other areas for light rays that are landing on these other areas? Less awesome. It's a perfect term. Okay. So, and here's what goes on with vision. We have about 100 degrees of vision. We have about 6 degrees that we can see really well. So if I focus on the clock in the back of the wall, I can see the clock very, very well, but everything outside of that is kind of fuzzy, kind of a little bit blurry. I can see movement that's happening in those areas, but I can't see the specifics of those areas. So I've got about 100 degrees of vision. I can see out here in my periphery, I can see forward, I can see low, I can see up, but there's a very, very small area of that visual field that I can actually see well. It's about 6 degrees. And that very, very small area is coming from light rays that are landing on the fovea. Okay? If I want to go from looking at the clock to looking at Brian, what I have to do is adjust the position of my head so that the light rays coming from Brian are the ones that are landing on the fovea, and everything else around that now in my periphery is kind of blurry, kind of fuzzy. So we have to adjust the position of our head as we focus from one thing to the next to get the light rays that we want to focus on coming into the fovea to be able to see them well. So we've got basically this circular structure here in the back of our eyes where we've got a high density of cones that is responsible for that 6 degrees, okay, that very, very small portion of our visual field that we can see well. So here's a human eye. I'm going to draw that to represent it for you. We've got this very, very small area where we're able to detect light rays very, very well. Can you guys see the blue in the back? It's kind of light. Sort of. Yeah, okay. Here's a crocodile eye. Okay. The shape of their macula is very different. Their macula, where they have a high density of cones, is shaped like this. Okay? What is that going to mean for the peripheral vision of a crocodile? It's good, which is kind of creepy, because I used to think that you could just like, you know, you hear they're really fast and they can outrun you short distances and they're ginormous, and I used to think, okay, you could like be on the side of a crocodile and you're totally fine, because they poke their heads up out of the water and, you know, they're looking forward, and we kind of anthropomorphize everything, right? We make it human and assume that their abilities are our abilities. Well, I can't see what's going on out here, but if I'm a crocodile, I've got this little slit of very, very good vision all the way out into my periphery, that I can see what's going on. And so I can be like this, and you can be a person sneaking up on me out here, and I can see you perfectly fine, even though I look like I'm looking out here, because my macula is not shaped like your macula, and I have very, very good peripheral vision, because light rays that are landing anywhere on here all the way out to the periphery are being picked up very, very well. So they have more like 100 degrees of good vision, where we have only 6 degrees. Where we can see well, and that just comes back to the shape of their macula, and where they have a very, very high density of photoreceptors located. All right. So maybe one last slide before we go today. So again, something that you may or may not have talked about in lab, but leads into some stuff that I want to get into when we start talking about refractory issues. And that is that the internal eye itself is divided into a couple of different segments, or a couple of different chambers. The first of these is known as the posterior segment, so it's basically behind the lens. This area right here, this open space, is that posterior segment or posterior chamber. It is filled with a relatively thick gelatinous fluid, which is known as vitreous humor. And vitreous humor is extremely important for at least a couple of reasons. First of all, the vitreous humor is going to pressurize the eye. And because it's pressurizing the eye, it's going to help the eye to maintain its shape. We need eyes that are almost perfectly spherical for proper vision. If our eyes start to become a little bit oblong in one direction or the other, that really interferes with our ability to see. So vitreous humor actually pressurizes the eye to some extent, helps it to maintain its shape so that light rays can pass through it properly and can be projected on the retina properly. The other thing that this vitreous humor does in pressurizing the back of the eye is it keeps the retina pushed up against the back of the eye. And the reason that's important is because the only place where the retina is actually physically attached is right here in the optic disc, where the optic nerve is exiting out of the back of the eye. In all of these other places, it's just a layer that kind of sits over the back of the eye itself. That's why when you did your eyeball dissections in lab, you may have had a little bit of a problem finding the retina, because as soon as you poke into that posterior segment and the fluid starts to leak out, you've now depressurized the eye and the retina falls. And usually what you end up with when you get done is this little globbly, gooky looking thing sitting right here in the bottom, which is your fallen retina. Having your retina fall is something that happens sometimes to humans as well. And people that have had that happen describe it as kind of like a black curtain falling over their eye, because as that retina falls, you lose the ability to respond to light rays that are being projected on the back of the eye. So anything that depressurizes the eye can cause the retina to fall, or anything that changes pressure. I've known people that have had it happen because they got shot in the eye with a paintball. I know people that have had it happen because they got poked in the eye playing basketball. I know somebody that had it happen because they were lifting something heavy, okay, and they were straining, and that caused their retina to fall. It's something that typically can be treated. There's laser treatments and surgical treatments to get the retina back up into the position where it needs to be. An exception to this is if it's fallen and it has torn, okay, because if it's torn and it's been obliterated into a whole bunch of little pieces, it's really hard to get those pieces back up where they need to go. And for people that have had it torn and maybe obliterated, usually what happens is they just lose a part of their vision. Okay, whatever piece was torn that's been ripped up into shreds, they can't get that back into place. It doesn't respond anymore, you know, and maybe that third of their vision or so is just gone. It's black because they don't have photoreceptors to pick up light rays that are coming in from those areas. Let's go ahead, we'll leave off there, we'll come back next time we meet, talk about the anterior segment, get finished up with vision, and then definitely we'll be moving into chapters